Okay, so my name is Veronica. I'm a biology and I'm actually a psychology teacher as well. So if you continue to do TSFX through the year, you might see a lot of me. I take the master classes usually on a Sunday and as many lectures as I can. I'm a teacher at Lauriston Girls' School. And I'm also a VCAA assessor. So I'm going to be giving you all my hints and secret tips, secret assessor information that I've been collecting over the years. Okay, so that's my goal for today. This is a great lecture. It's an introduction to what you're going to be doing this year. So I think it's awesome that you do this preparation. And you get a good sort of grasp of the concepts before you start school. To start the lecture with some tips, okay? And I don't usually do this. This is the first time I've done this at the start of the year. I actually did this at the end of the year last year. So when we did the revisions just before the exam, which was I think October last year, I did this little spiel. And I got bombarded with messages. Some students were really angry. They're going, why doesn't anyone tell us this at the start of the year? So I'm going to be telling you this at the start of the year. So it's pretty general type stuff. And it's stuff that I've picked up from being a VCAA assessor. So yesterday I was telling the students that I've been assessing, I think, for about 10 years. Juliet, who'll be taking you for the next two days, is like deputy chief examiner, and she's been assessing a lot longer than me. So she will give you different tips, but I'm quite new. So 10 years is considered quite new, which is weird. And before that, I had these really smart, conscientious students, and they just weren't getting 50s, and I couldn't work out why. And it wasn't until I became an assessor that I realised, yes, the content is important, but also the mechanics of answering questions. So I just want to give you a few tips because you're going to have a SAC in about probably week 6. So your first biology SAC will be really quick. And a lot of your teachers sort of are preparing you for the exams. That's the purpose of SACs. So they will be expecting this from you. So I think it's much better to know it from the start than making mistakes as you go along and dropping marks. Okay, so here are my top tips that I've worked out. Number one is there's two types of questions, I guess, for the biology exam. You've got a new study design this year, but we've already heard from VCAA that the format is going to be the same as it has been in the past. Okay, so I'm just multiple choice. Okay, so it looks like you're going to have about 40 of those again. Now, the trick with multiple choice, some teachers love to do multiple choice on SACs, is to read very, very slowly and very, very mindfully and watch for the distractors. So a distractor is one word that will make the statement wrong. So the A, B, C, and D are the 4 statements. And often what VCAA do is sort of almost like trick your brain. It's not just VCAA, it's any person who writes a multiple choice question. So you'll see something that's familiar and your brain will be drawn to that, but the word before or after that might be wrong, if that makes sense. So you really need to read a lot more slowly than you're used to. Don't take a gamble on multiple choice. A lot of my students say, oh, multiple choice, I can just have a guess. And if it's in doubt, choose C and all those stories. But every time you get one wrong, that's about 3% of your grade, okay? As soon as you get 3 wrong, which is pretty easy out of 40, there goes a whole grade. That's 9%. So, 10% almost is a whole grade. So you will be dropping from an A+ to a B+, which you don't want to do. So every multiple choice, you need to read them very, very slowly, watch for distractors, and also have a time budget, okay? So sometimes they're quite tricky. And sometimes it could be as early on as question 2 on the exam. This actually happened on the psychology exam in 2019. So question 2 was really tricky. And so a lot of students spent 5 up to 10 minutes just on question 2, and therefore they didn't have enough time to finish the exam. If you can't answer that multiple choice within 1 or 2 minutes, move on and then come back to it, okay? You can't risk time on the exam. So that's multiple choice. I hope I'm not freaking you out this early in the year. Now, short answers are awesome in biology. They're, usually no more than 6 marks. If it's 6 marks, that's extreme. And usually a 6 marker is filling in a table. They're often 1 mark, 2 marks, 3 marks. And that's about it. Dot points are fine, so you can answer in dot points. But here's where the secret assessor stuff comes in. Okay, so I'm going to give you quite a few acronyms today. The first one is to make sure you fully understand the question. So you need to actually study the question before you put pen to paper. Okay, so remember, you need to start practicing these skills in the SAC. So I have a little acronym that I've come up with. It's BUGS. Now, that stands for box the command term. And we're going to practice doing this, but underline what you think the dot point is, the VCAA dot point. Glance at the marks and squiggle the scenario or the example. Okay. So the main command terms that you tend to get in a biology exam are describe, explain, and sometimes right at the end of the exam when it's got the harder question, you might see a justify. You might also get some kind of comparative command term like what is the difference? So I'll go state the difference. Or state the similarities. Sometimes you will get a drawing. Now, there's quite a few other command terms. If you're not too sure of those, if that's really weird to you, just Google biology command terms, and there's lots of tables out there. There's one I like in particular. It's actually for IB biology, which they have at my school as well. And it's a slide share. So it's one of the first ones that come up in Google. And that's a really good one because it actually has the command term and then a biology example. Okay, so what I found from being an assessor is students knew the content really, really well, but they didn't actually look at the question and didn't sort of analyse the question before they put pen to paper. Okay, so if it's describe, there's actually 3 kinds of describes. You get describe the structure, you get describe the role. Now, if it's a role question, that means what is its function? And what I find particularly early on in the SACs is that's where students drop marks. I'll have, describe the structure of transfer RNA, for example, and students will actually tell me what transfer RNA does, if that makes sense. So you have to be really, really careful that you're describing the right thing. The other thing you get to describe a lot are processes. Okay, and one thing I've learned about processes is you need a beginning, what starts it, what's happening in the middle, and what happens to finish it. And that's usually 1 mark each for the 3 marks. So there's a real formula that I've worked out for the way you can max out marks. So you can have 3 really, really good points about a process. Like one of the processes we're going to be doing today is transcription. For example, you could have some great processes there that you've explained really well, but they might be middle processes. You haven't actually told me what starts the process of transcription or what ends it. So therefore, you've lost 2 marks, even though you've got 3 really good marks. All right. If you get an explain, try and use the word so twice. And when we practice these, I'll be saying, okay, this is a so-so question. If it's justified, please make sure you give a reason. And usually when you give a reason, it's a reason to either support or refute. Okay, so they're sort of good words to use when you're justifying an answer. You say these results support this because, or these results refute these because. Okay, because it's a reason, you'd probably want to use the word because. If it's a different question, you need to use the word whereas. And if it's a similarity, both. Drawing questions, they're usually about 3 marks as well. Also make sure you always draw it in 2D and make sure your labels are non-ambiguous. Okay, so classic example, there might be a picture like this and it might be labeled a nucleus and a student might go like that, nucleus, okay? That is wrong. The arrow has to smack on the nucleus. So make sure your labels are non-ambiguous. And yeah, they're the main things there. Okay. Now, the scenario is really important. You can't give generic answers. So, for example, they're not going to say, give me the steps in transcription. If you don't know what transcription is yet, you've probably got no idea what I'm talking about. But they might talk about a particular enzyme, say tryptase, that was on an exam a couple of years ago. And so if you've just given a generic answer about, what happens in transcription and not made it specific to the scenario and mentioned the word tryptase, again, you won't get the marks, you'll get 0 marks. So you have to be very, very specific. So that's why the scenario is really, really important. That needs, if the molecule is named in the question, make sure you echo that back in the answer. Okay, so the other type of questions that you often get are data ones, and so that means you must use data from the graph or the table to back up. And my last bit of advice before we get into content is if it's a 1 mark question, and this was the biggest thing I think I learnt. Okay, so it might be, say, let me do an example of something quite easy. Hopefully you all know about, a setup of an experiment and how it's important to have an IV and DV and control variables. So if it's got like state a control variable, which is a really common one you get on a SAC or an exam, don't just use 1 word. And even if it's a content question, what I've learned is it's very rare to have a 1-word answer. So I used to teach my students, if it's 1 mark, 1 word, don't waffle because you might shoot yourself in the foot. You might give wrong information. But I'm finding now that it needs to be 2 words. So for example, in a control variable, you might say, I'm going to control the temperature, which is always problematic. Okay, the temperature of what? The temperature of the room, the environmental temperature. If you're doing something in a test tube, the test tube temperature. If it's even same size test tubes, a lot of students will just write the test tubes, same size test tubes. Okay, so always try and have 2 words, 2 or more words right from the get-go in a 1-mark question. Now, I've got a few other tips, but I don't want to overwhelm you. I just want to give you enough for the first SAC, okay? So the command term is going to be really, really important. Be very, very specific in the SAC. Don't just give a general answer about a process. Name the actual molecule. If it's a 1-mark question, make sure you give 2 words. All right, so next we will go to probably the most important page in the book, which is this page on page 4. So if you've got your highlighters ready, this is basically everything. I'm going to be doing up to here today, except Juliet is going to do this one tomorrow. So she's going to do this dot point and then all the other ones here and the rest over the next 2 days. Okay, so this area here is my area. Now, these dot points are, like I say, everything. One thing I picked up, another trick I picked up from being an assessor, is anytime they have the word including in any science, this is the same with psychology, you can replace that with only, all right? So these are summarized, but see here, there's the word including. So it means like including, you have to include all those, but you don't have to include anything else. So RNA processing in a eukaryotic cell, it's only in a eukaryotic cell. But transcription in prokaryotic and eukaryotic, translation in prokaryotic and eukaryotic, and just RNA processing in eukaryotes. Juliet, this is new to the study design. So it's actually a trip operon. So in the previous study design, which was last year and before that, I think it was 2017 onwards, it was the lac operon. So if you're buying notes from Year 12 students last year, I personally wouldn't bother. But if you do, they would probably have really good summaries on the lac operon. Don't even bother learning about the lac operon because you only have to know the trip operon. Now, also, they haven't got the word including here, but you need to know this process is the protein secretory pathway, but they've given you this heaps of organelles in the cell. So yesterday we sort of played a game where, it wasn't really a game, but everyone wrote 10 organelles found in the cytoplasm of a typical eukaryotic cell, okay? So there's lots of them. And the students who came yesterday for the prereq lecture had no trouble listing 10. But you only have to know these ones. Number 1, rough, okay? It doesn't say smooth, rough endoplasmic reticulum. And please never, ever, ever write ER. It has to be endoplasmic reticulum. 2, the Golgi apparatus. Then it's got associated vesicles. So that means transport and secretory. So there's 2 types of vesicles. And that is it. So there's only 3 organelles that you really need to know about. And notice how it says the role. So you need to be really, really clear. What do each of those 3 organelles, or 3.5, do when we're talking about proteins being secreted? The other and the final thing to point out here are definitions. Use these as your definitions. So, for example, the first topic we're going to be doing today is nucleic acids. And there might be because it's the first topic, number 1 SAC question your teacher might put is define nucleic acid. Okay, so have a think. What would you write for that? Most of you probably know what a nucleic acid is. How would you define it? So let's have a bit of thinking time. I'll just be quiet for a few seconds. Molecules. Oh, Damien, you're too good. Okay, I was hoping someone would write something like, oh, it's, an organic molecule that includes DNA or RNA or something like that. But Damien nailed it. You were nailing it yesterday too, Damien. So what was awesome about Damien's answer is this. Everything I highlight in yellow is a definition. So use the VCAA definition. Okay, so remember, I'm just doing the first part today, but it'd be a really good exercise before your SAC to do this for every single dot point. And Tara, that is such an awesome answer. And this is what I learned from being an assessor. Tara is exactly right. It is a polymer of nucleotide monomers. And I would definitely include that in your answer. But this is what VCAA has put in the study design. It's an information molecule that encode instructions for the synthesis of proteins. Okay, so so everything I highlight, think of this as a definition. The next one is awesome. This is actually a new one too, so I'll put a star next to it because if it's new, teachers usually like to be a little bit conscientious about putting it in the SAC, and it's usually on the exam. All right, so last year we learnt in the study design that the genetic code is just basically degenerate. Oh, sorry, I didn't mean to highlight this bit here. And if I can, maybe I'll go. Don't worry about this bit here. I'll just get a pen. Okay, but they've added to it this year. So when you define genetic code, they would want you to use those words, universal triplet code that is degenerate. Okay, so that's the definition for genetic code. If you have to talk about or label a gene, there's lots of different parts of a gene, but these are what they want you to label. I might not do that. I might do that in green, sorry, pink, because that's not really a definition. But if you had to draw a gene, there are 4 regions that they would expect you to label on the gene. Here's another definition of an amino acid. Okay, so this is sort of like what Tara's answer is, but it's the monomer of a polypeptide chain. That's what an amino acid is. What is the definition of proteins? Again, you might say it's a polymer of amino acids. Wrong, according to this. It's a diverse group of molecules that collectively make up an organism's proteome. And notice this is quite new as well. I'm really mucking up my colours here. I wanted to have yellow as my definitions. Here's an including. So that means the case study they want you to know about is enzymes. So enzymes is going to be your big case study for a protein. Okay. All right. So hopefully that's giving you a little bit of the area. It doesn't look like much. So I've actually only got, like I said, I'm only doing up to page 64 today, but I've got the foundational stuff. So I need to do it in a lot of depth for you guys. So even though it's not a lot of VCAA dot points, it's going to be quite intense, and we're just going to make sure you've got a really good understanding.